So today we're talking about the IonTorrent system. This is by Carly McAllister, Maggie Williams, and Ali Brem. IonTorrent sequencing can be used in the fields of disease research, cancer research, reproductive health research, and human identification. Some of the key applications for the torrent system is chip sequencing, which actually looks at the protein interaction with DNA, exome sequencing, gene expression sequencing, de novo sequencing, small RNA sequencing, whole transcriptome sequencing, viral and bacterial typing, and drug resistance sequencing. So on this slide, we include a picture of RNA transcription because unlike other sequencing tools, IonTorrent can look at the molecular mechanisms of gene transcription while cataloging complete sets of transcripts while quantifying changing expression levels. This can also be used to discover novel sequencings, exomes, and biomarkers. Conventional gene expression profiling methods include hybridization-based or microarray and amplification-based quantitative reverse transcriptase PCR technologies. However, both approaches have limitations. Neither method enables absolute quantification of transcripts. Hybridization-based approaches suffer from compressed fold change accuracy and low sensitivity. The workflow associated with amplification-based methods does not enable parallel analysis of large number of genes, for example, as in a pathway or as in a disease. IonTorrent RNA sequencing provides a representation of absolute expression and can identify and characterize low abundance transcripts and sequence the whole transcriptomes. The machine that is used is actually benchtop, and it has an output up to 10 gigabytes, and it can do total reads of 60 to 80 millions while running only 2 to 4 hours, which is much shorter than traditional next-gen sequencing. The way this machine works is it uses a small semiconductor chip that contains millions of tiny wells that can capture chemical information. In order to read the DNA or RNA, it's fragmented and then mixed with small beads. One fragment of DNA or RNA attaches per bead. An emulsion PCR is used to amplify the fragment of DNA or RNA until it completely covers the bead. Once the bead is completely covered, each bead with its corresponding fragment is put into a well.
Once the chip is loaded with the beads, the wells are flooded with DNA nucleotides one at a time, every 15 seconds as seen in the image. So you can see we start with adenine flooding the well for 15 seconds, then thymine, guanine, and cytosine. The cycle repeats itself until the DNA is fully sequenced. When a nucleotide is incorporated, a positive hydrogen ion is released. An ion-sensitive layer underneath the chip reads that pH change as a voltage. The level of voltage determines which base is added, and the software calls out that base. When comparing IonTorrent to Illumina, it is much cheaper to use IonTorrent at only $50K compared to about $125K. It can only sequence about 1 gigabase compared to Illumina, which can do 1.5 to 2 gigabases. The cost per gigabase is about the same at about $500. Runtime for IonTorrent is only about 2 hours compared to Illumina at about 27 hours. However, the observed error rate is much higher at about 1.7% compared to Illumina with only about 0.8% error rate. The read length for IonTorrent is about 200 base pairs, while the read length for Illumina is only up to about 150 base pairs. Insert size for IonTorrent is much smaller at only up to 250 bases, where Illumina you have an insert up to 700 bases. The scientific article we looked at bacterial community shifts of patients with periodontitis and 2 different treatment options. The 2 treatment options were scaling and root planing to remove biofilm, and then scaling and root planing plus antibiotics. The use of IonTorrent allowed the gingival bacteria to be sequenced both before and after the selected treatments to determine the efficacy. This image shows the prominence of each bacterial genus in the controls and each experiment. Presence of the bacterial genus was determined via IonTorrent gene sequencing, looking at the 16S rRNA subunit.